Guru sir,
Please proceed. It is audible. It is audible. I can able to see the slide. Good
okay,
afternoon. Good afternoon. Please proceed.
yeah.
Okay.
Yeah.
So, good afternoon for our external examiner, Guru sir Satyendran from University of Hyderabad. Um, I also welcome to all the participants through online and offline in the conference room. Actually, this is the Google meeting for the Viva was examination for my student, Kakoli Malakar. She submitted their thesis a couple of months back. We received all the three reports from the external examiners. So based on that, the controller of examination suggested me to conduct the Viva's examination. So that's why I arranged for this Google Meet. So in this ground, I asked Kakoli Malaka to present her slides, maybe 30 minutes, like because the external examiner attending on the MT conference in IISc Bangalore. So that's 30 to 40 minutes. You can finish your presentation. That will be helpful for you. Okay. I invite Kakoli Malaka to present that thesis.
Thank you, sir. Good afternoon, respected professor and all the participants present here. Today, I, Kakuli Marakar, going to present my PhD work on the topic synthesis, characterization, and redox behavior of transition metal complexes of certain dithiocarbazates ligands. So this is my thesis overview. Here I will be discussing about in chapter, I will be discussing these things. In chapter 1, I will discuss about general introduction of the metallodrugs. In chapter 2, here I will discuss 6 new type of dithiocarbaxate ligand. In chapter 3, it's all about heteroleptic complex. Chapter 4 deals with some homoleptic complex. And in chapter 5, we will be discussing about some bulky complexes, 1:2 bulky complexes. And in chapter 6, we will be discussing monomeric and dimeric complexes. So chapter 1, metallodrugs and its potential application. Now the question comes, what is metallodrug? Simply the drug which contain metal. So why do we use metallodrug? Because since extensive use has been observed in our human, extensive use of the metal has been observed in our human body, that's why therapeutics comes with the idea of the metallodrugs. So different scientists has classified metal drugs in a different way. Here we have followed one standard paper, and here we have classified in a 7 way, like inert metal complex, reactive metal complex, metal complex fragments, complex that deliver an active ligand, complex that deliver an active metal, radiation sensing complex, and radiotherapeutic complex. So condition for designing metal drug. Before designing, we have to know how the metal drug works. Actually, when we consume the metal drug orally or in other way, then what happens? It reacts with the body fluids, with blood, and then directed to the targeted biological targeted, and then deactivate the targeted and fragmented and then excreted way. What things we have to keep in the mind when we design for metalloducts? This is types of the ligands, which type of ligand you are using, like nitrogen, sulfur, which kind of donor groups are present, then outer sphere reaction depending on the presence of the different heteroatom, outer sphere interaction will be different, then ligand exchange rate, toxicity, steric and electronic feature. And for metal one, we have to keep in the mind chelating ability, you are using small metal or bigger metal, and which kind of ligand you are using, depending on that it will affect. Then coordination number, if you use a small metal with fast transition series, then coordination number will be less, but if you use a platinum or palladium, then coordination number will be greater. Then choice of the metal, of course, which kind of metal you are using, then geometry also, and oxidation state, it has a very important role in our biological body, so oxidation state also play an important role. Example of metal drugs, this is the salvarsan, which is the first metal drug. It was in 1916, it was invented by Paul, and this has been used for the treating of syphilis disease. And this is some gold metal drug which is used for the treatment of the arthritis. And these are very famous drugs, cisplatin, niraplatin. This is used for the treatment of the cancer. Characteristic of dithiocarbaxate. Actually, nowadays thousands of ligands are available. So why we are using the dithiocarbaxate? Because there is certain property with the dithiocarbazates. What is so special? Like as you can see, this is it can especially the dithiocarbazates. It can exist in two form, like thione form and thiol form. So it can bind in a both form also, and two nitrogen and two sulfur are present also. So depending on that, the solubility issue will be solved, so it can be soluble very easily. And within this R group, you can incorporate different kind of heterocycle and different kind of donor atom in R group, and depending on that, it can chelate in a different way. Beside that, the conformer also, as you can see, it can exist as cis 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 trans cis and trans conformer. And if you depending on the conformer, it's capable to do hydrogen bonding also, which is a very useful criteria for a drug. Now the question is, can you use this complex of the ligand as drugs? Of course we can use, because when you incorporate a metal in the ligand, then solubility also increases, then we will get more structural diversity as well as chelating properties. Importance of the copper complexes. So these are the copper complexes which has been used for the cancer treatment. These are in clinical stage. And this semicarbazone, which is, you can see, the sister of the dithiocarbazates, it also goes to the clinical trial. Beside that, copper complexes, it has been used as antioxidant, anti-cancer, antifungal, antibacterial, and catalytic activity. And it is present in different type of enzyme also. And another advantage is that it has lower toxicity, and copper is cheaper compared to the platinum and other metal. And facile synthesis, you can easily make the metal complexes, and you can the yield is generally very high. So in literature, there are different type of S substituted dithiocarbazates is used. So as you can see, these are reported as per now for S substituted dithiocarbazates. So anti-cancer active dithiocarbazates. People are trying to discover some anti-cancer active dithiocarbazates, and here there is some literature where this zinc complex and mixed metal complex of vanadium, then copper complex, dimeric copper complex, this all complex shows very effective activity towards the cancer cell. And apart from the cancer activity, it is also can be used for antimicrobial catalytic activity and BSI activity. For antimicrobial activity, in paper it's shown that that's square planar one, it reacts efficiently other than this octahedral and tetrahedral geometry. And catalytic activity also has been shown by this metal complexes. BSA binding is less explored, only one paper is till now reported. So here is it. Scope of the work. As we know, the biological property is changed with the slight change in the structure of the ligand. So this ligand could be a efficient choice, and this ligand can use as all this field from anti-cancer to dyes and pigments. And before I show you that, the introduction of dithiocarbazates with different functional group or use of the other transition metal tune the photophysical and redox properties. Objective. Objective of this thesis was to synthesis and characterize of bioactive ship-based bi and tridentate ligands derived from S-methyl benzyl dithiocarbazate and copper complexes. And we have studied interaction studies of the complex with DNA and BSA protein. Then we have studied the anti-cancer activity and illustrated their structure activity relationship. And we also studied the mechanism of the cell death with a different kind of biomedical techniques. Chapter 2. Here I will be discussing I will be discussing about 6 new type of ligands. So here we have used benzimidazole, imidazole, and pyridine. So why benzimidazole and imidazole? Generally, these are present in different type of enzyme, and this type of compound has been used from antiviral to anti-cancer activity. Yeah, in a nutshell, for many biomedical application, this has been used. And why pyrrole? Because pyrrole is a toxic molecule. We use pyrrole because it's a planar system, and because of the planar system there is extensive delocalization of the electron, and for that it has a very small homologue gap, and that gives it very strong donor property and makes it a very good photoluminescence compound, and makes it a suitable fluorophore unit to bind the BSA. So this is the structure of the ligand. This is the imidazole with S-methyl, imidazole with S-benzyl. This is benzimidazole with S-methyl and benzimidazole with S-benzyl. And this is with pyridine with S-methyl and pyridine with S-benzyl. So these are the NMR spectra. Here, this 10.36, this proton is for NH, and this 8.81, this is for this CH, means azomethane CH, and all these are for aromatic compound. This is the solvent peak, and for 2.69, this is the aliphatic proton SCH3. For carbon 1, this is 198, this is for C double bond S, and 146, this is for C double bond N. These all are for aromatic compound, and 17.38, this is for SCH3. So mass spectra, this is, it comes with the combination of the proton, and this is the IR spectra as shown. This 3092 is for NH, and 1583 for CN, 1145 for CS, and 1046 for NN, and 956 in the range, this is for CS. So this is the structure of the L1 crystallographic structure. We have drawn the crystal from the methanol, and this comes with the orthorhombic crystal system with space group P212121. And if you see the crystal system, it comes in a helical structure. So this is the benzimidazole crystal. We have drawn the crystal from methanol only. The crystal system is monoclinic with space group P21C, and it comes in a straight way. And if you see the C double bond N, we can see this is a trans structure. The two bulky groups are apart from each other, gives it the most stable structure, and that is true for the previous crystal structure also. So this is the hydrogen bonding. Both compounds are capable of doing hydrogen bonding. This is intermolecular hydrogen bonding. So all the details of the hydrogen bonding is given here. So for the thermogravimetric studies, as you can see, L1 and L2, it contains imidazole and very small molecule, and at 193 and at 192 it decomposes only. But for the other two molecule, that is the benzimidazole one, it decomposes in a two step. In 196 to 220 is for SCH3 fast weight loss, and for the second weight loss, the compound decomposes at 220 to 377, and 168 to 207 according to literature survey is for S-benzyl, and rest of the molecule decomposes at 207 to 309. This is the UV structure. As you can see, 320 to 400 nanometer. This is because of the pi to pi star and n to pi star transition. This is because of the imidazole and benzimidazole which is present in the ligand. So BSA binding study. BSA is a big molecule with 583 amino acids and has 17 cysteine residue. It has 55% alpha helix structure, and remaining structure is beta. This compound is fluorescence active and can be divided in a three domain, and every domain can be divided in two subdomain. This is mainly fluorescence active due to the tryptophan, tyrosine, and phenylalanine. And if you can see the blue and red shift with your compound, that means it changes is the hydrophobicity of the microenvironment around the tryptophan residue. So interaction of the BSA with the ligands. So what we have done, we have taken that 5 micromolar of BSA, and then keeping the volume constant, we add different concentration like 1 micromolar, 2 micromolar concentration of this metal. Then we can see that after taking the fluorescence, that intensity decreases. What does that imply? That implies that our compound is binding with the BSA. So after that, we have done the absorbance also. This is the means opposite process, reverse process, that can so that you can see the absorbance increases. So from there, we have plotted that Stern-Volmer plot and that double logarithmic plot, and from here we have evaluated that KSB, Stern-Volmer constant, KQ, rate quenching constant, binding constant, then binding site, and the free energy. So as you can see that for the L6, the n value is 1.16, which is the highest. N is the binding site, means in how many sites are binding with the BSA. So and the G value, all the G value are negatives, implies that it's spontaneous process. And as you can see, it is in the range of 10 to the power 13. That means it follows the static mechanism. And all the binding, this is the sufficient binding, so you can tell that all compounds are potent quencher. So this is the in vitro cytotoxic assay. It has been evaluated against the cancer cell, squamous cell lung carcinoma H520. From here, we have plotted concentration versus the percentage of inhibition. Here you can see that in higher region, L1 predominates, but in case of here, you can see that in lower region, L3 predominates, whereas in in the case of L4 predominates in the higher region. And from here, at the rate of 50, we have evaluated the IC50 value. From here we can see L1 has the lowest IC50 value. The lowest the IC50 value means the compound is more biologically active, means it has the more ability to kill the cancer cell. So according to the cytotoxic efficiency, we can see L1 is more efficient and L4 is the least. So acridine orange and ethidium bromide staining, these are basically dye. Acridine orange, it gives green fluorescence with the live cell, and ethidium bromide, it gives orange fluorescence with dead cell, and it doesn't react with the live cell. So as you can see, this is the control in DMSO, and from here you can see that L3 and L4, these two are mostly means this is a qualitative process. We can't say like that, but we can just give an we can just take an idea that L3 and L4 here is mostly kill the cancer cell. This is the hair staining. From here also, as you can see, they are also most compound. This is the morphology changes for the L3 and L4 most, so we can say that L3 and L4 are most effective. So we have optimized the geometry with the Gaussian software at the temperature 298 Kelvin, that is room temperature. As you can see that if you compare the energy gap here, L5 and L6, that is the pyridine derivative, they have the less homolymo gap because they have the extensive conjugation. Pyridine is, as you can see, pyridine is a very planar molecule, and there is a good delocalization of the electron. That's why that energy gap is very less, and energy gap is very less means these compounds are very active, and we can see from the BSA binding studies also, this shows the highest value. So conclusion of the chapter, we have successfully synthesized and characterized 6 novel compounds. Then L1 crystallizes as orthorhombic and L3 as monoclinic. Then all compounds are potent quencher, and L1 shows lowest and LC show highest binding interaction with the BSA, because L1 is very small molecule and L1 has lowest conjugation, but L6 has highest conjugation. That's why it shows highest binding interaction. And L2 and L4 shows low toxicity and anti-proliferative activity against the cancer cell due to the presence of the benzyl acetyl group, which is evident from the literature. All compounds can be used as potential drug. So chapter 3, here I will be discussing about 8 new heteroleptic complexes. So with the dithiocarbazates, we have here incorporate with 110 phenanthroline. First, let me tell you what is heteroleptic complex. Different types of ligand, they bind together with the same metal ions. That is heteroleptic complex. And if you incorporate 2 types of ligand with the same metal complex, you will get more structural diversity and molecular design. And it has a different type of application, from catalytic activity to anti-cancer activity. Now, why do we choose 110 phenanthroline? Because 110 phenanthroline is a planar molecule and can interact with the DNA very efficiently. Beside of that, this also inhibitor of several peptides, especially the zinc one, and with the lower affinity of the calcium. And the inhibition of the enzyme occurs by the removal of the chelation of the metal ion for the catalytic activity, leaving an inactive apoenzyme. So this is the general synthesis for the tridentate ligands. Here first we use the pyridine with hydrogen, then pyridine with methyl, pyridine with phenyl, then quinoline, then quinazoline, then imidazole, then benzimetazole, then N,N-dimethyl. So this is the synthetic procedure of the metal complex. We have taken 1:1 with the copper perchlorate salt and obtained this complex. So these are the structure of the complexes, the 8 complexes. So NMR of the ligands, as you can see, 10.05, this is for the NH, 9.87, this is for the benzimetazole NH, this 10.05 is,
Yeah, yeah. You can skip this one. You can skip
okay, okay,
this
sir.
one. Okay. Yeah, yeah. This
I
also skip.
also,
Please,
okay,
please,
sir.
please. Um,
You
skip,
people, sir,
please, 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 please,
this is the mass
please,
spec.
please. Yeah, yeah.
Okay.
Skip, skip, skip.
This one also I'll skip, sir.
Yeah, yeah, yeah. Yeah.
So
Now please proceed.
then,
Go ahead. Go ahead. Go ahead.
okay, sir. This is the crystallographic studies for the complex one. As you can see, the tau value is 0.28. So as it closes to 0, that's why we can say that it shows the distorted square pyramidal geometry. And distorted, that could be justified because of the bite angle of the ligand. So these are the selected bond angles and these are the selected bond lengths, and we evaluated the tau value with this formula beta minus alpha, where beta and alpha are the biggest angle divided by 60 degree, and beta should be greater than the alpha. This is the triclinic crystal system with P1, and we got other crystal system also, and mostly all of them are distorted square planar, square pyramidal, sorry, distorted square pyramidal geometry. So this for the 5 to 7, or mostly they are triclinic and monoclinic. And DNA binding studies, as you can see that we have done the same thing which we have done for the BSA. We have taken different concentration of the metal complexes and DNA 5 micromolar
Yeah,
of the
yeah, yeah,
DNA
yeah, yeah. The procedure will be same, na? Just conclude this chapter, that core thing in this chapter.
okay, sir. Actually, yeah, this absorbance increases, and all are means from here also we can say that the DNA binding studies all are the potent quencher of the DNA also. So it could be used as the anti-cancer drug. So psychotoxicity also, as you can see, this is for the complex 5 for 24 hours and 48 hours. And here we have seen that the killing ability of the cancer cell, 5 is the most efficient and 7 is the least efficient. And 5, it contains quinoline, 2 nitrogen. Because of that, it could be possible it is showing the efficient activity. And all the complexes, it kills the cancer cell, but for healthy cell it doesn't have any effect. And all the complex, it shows more IC50 value than the cisplatin. So this is the ROS study. From here also we can see that the ROS means the more the ROS generation, the compound is more efficient. From here you can see that the 5 is the more efficient, as the ROS generation is